Welcome, everyone. Thank you for joining us today for our final CE webinar in the MyDoc webinar series for 2024. It's wonderful to see such a full house today, and I'm so glad you could join us. We're excited to announce that we'll be launching more CE webinars in 2025, so please be sure to subscribe to our newsletter to stay updated on upcoming events. We are wrapping up this year on a high note with our fantastic speaker, Dr. Krista Keller from UC Davis. And for those of you joining us for the first time, here's just a brief introduction into MyDoc. We are an independent diagnostic lab. We utilize next-generation DNA sequencing to identify all pathogens in your samples, which includes all aerobic and anaerobic bacteria, as well as fungi, and antibiotic sensitivities to 43 different antibiotics and 8 antifungals. Results are available in just 2 to 3 business days. Coming soon, we will also be launching tests for DNA viruses and parasites, and we'll be reducing our turnaround time to just 24 hours. All of our samples include free shipping and free consultation. At the end of this lecture, you will see a QR code to download your CE certificate. If you are unable to download it from there, don't worry about it. You can reach out to our customer service team, and we will email you a PDF copy of your certificate. If you have any questions during the lecture from Dr. Keller, please feel free to ask your questions in the chat, and we will go through all of the questions in the Q&A session at the end of the presentation. And now it's my pleasure to introduce our speaker, Dr. Krista Keller. Dr. Keller is a board-certified specialist in zoological medicine, and she's currently Associate Professor of Clinical Zoological Companion Animal Medicine and Surgery at the University of California, Davis. In this role, she teaches the next generation of amazing veterinarians and zoological medicine specialists while providing clinical care for birds, reptiles, amphibians, and mammals. Her research is focused on mycotic infections of reptiles, whether it's supposed to be about the understanding of mechanisms of disease manifestation or exploring novel diagnostic therapeutic options. And with that, Krista, please take it away.
Thanks, Janina, and hello, everybody. Very, very excited to be here with a great audience. I was kind of running through several of the names, and I see some friends are definitely in the audience, so thanks for being here. Anyone who knows me knows that I can talk for well over an hour about fungus and reptiles, but Janina and the team said keep it to an hour, Krista. So what we're really going to focus on today is going to be the diagnostic criteria and what have we learned with a recent study that we've done out of my lab about which of the diagnostics, including the MyDog NGS technology, you can utilize to diagnose this fungus. So let's take it away. All good lectures have learning outcomes. Hopefully at the end of this, you all never are going to say the word can be again. You're going to be suspicious when people say the words yellow fungus disease. You'll be well-versed in recognizing that clinical presentation, and then you're going to understand some of those advantages and disadvantages of the available diagnostics, including NGS through MyDog. So let's get rolling with why can't we say can be? And this is a sticker that I had made because I am passionate about you can't say can be. So just to kind of go back into the past, we're talking 25 years ago, we discovered Chrysosporium anamorph of Menisiopsis varizii, that's CANV. At the time, it was an emerging global fungal pathogen that caused mostly cutaneous mycoses, sometimes more systemic as well. We knew it was challenging to treat and that really all reptilian taxa could be affected. Fast forward, now we're about 10 years ago, we had molecular characterization. So all of these can be isolates were grouped together because as I will show you, when you grow up these pathogens in the lab, they all really look the same. Their colony morphology is the same. When you look at them under the microscope, very, very subtle differences. But once we look at the genome, that's when we were able to divide this group of CANV into 3 separate genera. That's Euphidiomyces, Menisiopsis, and Paranenisiopsis. Again, this was over 10 years ago now. In 2019, there was a new player on the block, which is Amidomyces testivorians as well. But we're here today talking about some of the fungal pathogens that are going to cause this yellow fungus disease, which will break apart that terminology as well, but this lizard dermatomycosis. What we know is that N. goroi has been proven to cause this presentation in bearded dragons, and Nanisiopsis dermatitidis is proven to cause this presentation in chameleons. However, any day we can find any number of Nanisiopsis isolates plus Parananisiopsis also being able to cause these lesions. And then if you experimentally are infecting bearded dragons, you can cause a very similar dermatomycosis by infecting them with Oophidiomyces. So this is the fungal alphabet soup of, what's going on with lizard mycoses right now. And a big reason why we shouldn't use the terminology CANV. It's just really, really outdated, and we have newer information. So that brings us to when we have a lizard that we're suspicious has dermatomycosis, that we're probably targeting a diagnosis of Nanisiopsis or Parananisiopsis. Both of these fungi are onogenalian fungi, which means that they have a lot of keratinases inherent in them. And those keratinases allowed them to invade, the dermatologic system. And not only are they associated with reptile mycoses, but there's several Nanisiopsis isolates that have caused disease in humans as well. So I told you part of the reason why CANV became CANV was that when we grew up, all of these fungal agents, they really look similar on agar. And so each of these looks maybe a little bit different. And that's just because of, these concentric growth rings may look a little bit different. But in reality, how these colonies look is very, very similar. They're white. They're fluffy, they're powdery, they're dense. And so we can't morphologically say that one is one species versus one is another. And hopefully you can see over here on the right side of the screen, these are Parananisiopsis. So here we have two genera represented that really all have similarities in colony morphology. Further, if you take a swab of those beautiful colonies of fungus and you put them under the microscope, there are subtle differences to the way the hyphae will branch, and maybe the shape, a little bit of the shape and size of these Ailuroconidia, Atheroconidia, but these are so subtle that there's a lot of overlap, and this also isn't going to be a way that we can ID the fungi. So this really pushes us towards why those molecular technique papers were required 10 years ago to say that we have different genera here, and why, and we're thinking about moving towards identifying which fungal pathogen is infecting our patient, why we need some molecular techniques. So when we think about Nanisiopsis and Parananisiopsis and a Venn diagram of which hosts do they infect, hopefully you can see here, this is what represents natural infections. We have a lot more host dynamicism with the Nanisiopsis. When we add in experimental infections, and I apologize for the overlap, we see that snakes can be infected on both sides with both Nanisiopsis and Parananisiopsis. So that just helps us identify, we're not saying CANV anymore. I know some of you might be saying CANV, but we do have to break it, right? It can help us go into the future by knowing, which of the fungal genera are we really considering. The next big important point is that not all fungus is yellow. So this terminology of yellow fungus disease is super ingrained in the literature. It is so ingrained in the literature and what we say that actually the paper proving Koch's postulates for Nanisiopsis goroi as the cause of dermatomycosis in bearded dragons used the term yellow fungus disease to describe this presentation. And I put up this great artistic pic that I made myself to really kind of highlight what is the classic clinical presentation of this yellow fungus disease or dermatomycosis. And hopefully you can see here, it's facial, it's yellow, it's crusty, it's erosive to ulcerative. We have a lot of loss on the normal beard scale that would be there. So this is the classic presentation. But it is by far not the only presentation. So here are two bearded dragons, the ventrum of a bearded dragon, more the tail of another bearded dragon. Lesions are not just on the head and face. They can be all over the body at any dermatologic site, including in a whole bunch of stuck shed around this very abnormal scoliotic tail. So don't stick with that fact that it has to be around the face. Sorry, the color scheme on this slide came out a little bit weird. Just to point out, and I'm going to show this a little bit to you guys later, we have a recent study where we infected the bearded dragons, and actually their most prominent feature after being infected with N. gori was that they had femoral pore infection. That was the first site and the most dramatic site that they had a dermatomycosis. So truly, any cutaneous location can be infected. Lesions should not be thought to be limited to the skin or cutaneous sites. Here's a bearded dragon who did have cutaneous lesions, but also had these granulomatous lesions that were filled with fungi, Nenisiopsis, inside this bearded dragon's liver. And I have published on a case of a bearded dragon who had internal lesions without any cutaneous lesions at the time of her euthanasia. And then here's the big crux here, that lesions are not always yellow. Again, I'm sorry for some of the color here. This says clinical presentation. Here is a bearded dragon named Frankie. This dark kind of black melanized colored area is her site of infection with proven N. gori infection. This bearded dragon over here, unfortunately, his biopsy site dehisced, which is why there's a big open lesion. But this hard, kind of brown to black, almost Eschar looking area is also proven Nenisiopsis infection. So, here it is. We've been calling this yellow fungus disease. And the reason I harp on this, that maybe we should move away from this terminology, is that I feel like we sometimes can put on our blinders, right? We can say, oh, well, yes, this is the yellow crusty lesion. This could be nenisiomycosis or paranenisiomycosis. But we also want to keep our eyes open to maybe some of these less traditional, less classic lesions. So don't let the non-yellow lesions feel excluded. All of these fungal lesions can come in any color or variety at any site on the body. All right, and then really the crux of what we want to talk about today is that as veterinarians, as people trying to get a diagnosis for our clients, sometimes it feels like there's a lot of diagnostic options, but how to wade through, what is the best use of my client's money in order to detect this fungus or detect something out or rule it in or rule it out, it can be a little bit overwhelming. So hopefully kind of walking through this in a theoretical way will be helpful. So when I have a sick animal that presents to me, and lots of times now they come on referral for yellow fungus disease, but maybe the client is saying, hey, there's a skin lesion. And so my spidey senses are tickling for maybe this is a cutaneous mycosis. There are kind of three big categories I like to consider of diagnostics. The first one is systemic health, but also detection of fungus. Just is there fungus there as a binary question, yes or no? And then if the answer is yes, can we confirm that fungal identity by saying this is either Nenisiopsis or paranenisiopsis? And sometimes being able to say deeper than that. This is Nenisiopsis barbate versus N. gori. So we're not going to dive into bearded dragon health or chameleon health or individual animal health. There's a lot that we can do to pursue, how is this animals' general health? Is there something else going on? Do they have a reason why they're going to succumb to infection? And the big one I just want to point out here is imaging. I showed you a picture of a bearded dragon who died and had awful fungal granulomas in their liver. If we're just focused on the cutaneous manifestation of these microbes that are there, and we don't go diving a little bit deeper, we're going to miss some of these more systemic manifestations. And I suspect we are missing some of those systemic manifestations. So just keep that in the back of your mind, that a lot of these animals could benefit from having some imaging, whether that's even basically starting with CT could help you look at lungs, but we're probably going to need ultrasound and radiographs can help you look at lungs, but we're probably going to need CT or ultrasound to help us evaluate the liver and other organs, soft tissue organs in the coelom. So, I said fungal diagnostics, we have these kind of two broad categories. There are fungal detection tests. You see a lesion, a wound on an animal. There's lots of things that we can do to say, is fungus there, yes or no? Those are what I call fungal detection tests. And then there are fungal identification tests, and that's where we can really get, is this Nenisiopsis or Paranenisiopsis or something else? And, can we get down to a species level there? So let's just kind of walk through what are the pros and cons of each of these. So here are the diagnostics that can help us detect for the presence of fungus. And the first one is cytology. This is in office lots of times, or if it's not, I'd push you to try to get in office. You can take the crust and put the kind of moist part of the crust on a slide for an impression smear. And what we're looking for are inflammatory cells. We're going to look for secondary bacterial infection, but we're really going to look for Canidia and hyphae. And a lot of people feel like, oh, I'm not sure that I can diagnose that. You can diagnose that, and we can teach our technicians to diagnose this for sure. We can pick up that there is fungus there. So these are from the bearded dragons that we recently infected. We're going to talk about them. This right here, you can see the hyphae kind of a little undulating over here on A, more straight here. This is just with a right Giemsa, which is going to approximate what you can do in your office with a diff quick. On the subsection B, hopefully you can see here's a red blood cell, and then we have a lot of background kind of material and some naked nuclei, but here are these Canidia, okay? And we can key in that these are probably fungal organisms because a lot of fungal organisms are going to have negative staining kind of regions right around the fungal structure. And so very easily we could pick up, I wouldn't know that this was Angoroi, but being able to say, hey, there's fungus. And based upon the host species and the clinical presentation, I'm concerned with a Nenisiopsis or a Paranenisiopsis in this case. So just a quick case just to kind of show you an example of how cytology can be helpful. We're talking about fungus, because that's what I love. But of course, there are other differentials for skin lesions. So here's a bearded dragon with some black skin lesions, especially if they're crusty. That might be indicative that there's a fungal microbe. This animal was treated for 6 weeks with voriconazole, no improvement. And that treatment was based upon a presumptive diagnosis of yellow fungus disease. But when this was aspirated, it was actually a round cell neoplasia and was more in line with being a chromatophoroma. So just another way that we can use cytology in office to help us navigate diagnoses in our patients. Here's another case. This is River. He was a patient of mine. He was diagnosed with meningiomycosis molecularly. He was started on terbinafine therapy. And at that time, cytology really supported that molecular diagnosis because we saw fungal hyphae and Canidia. He initially improved upon recheck. And so here he is in one of his rechecks. He had one of these classic facial lesions that I've shown you a picture of. But then 6 weeks later, the facial wound was worsening and he was still taking his terbinafine, and we did a repeat cytology and we saw that there's a really severe monomorphic gram-positive rod overgrowth that was there. And so we were able to start him on an appropriate antibiotic regime on top of his antifungal regime and get that wound back into a state of getting towards healing. So just another example of how cytology can really be helpful. That next class of how to evaluate for the presence of fungus, just binary, is there fungus, yes or no, is fungal culture. This can be from a lesion swab. This can be from a biopsy sample. There's some discussion about, do we need to send to specialized labs? This is, and I'll show you some data around this. This is a fungus that grows pretty readily, pretty easily. Other reptile fungal agents do not have that same growth characteristic. So I can't comment on, a large lab's capability, like a large commercial lab's capability on growing this up. But I have heard, kind of from the field, that people say that they have had challenges with some of the commercial labs. But a fungal culture can give you, this is this is positive for fungus. I've shown you lots of pictures. Here's a truly beautiful one of the fluffy, hyphal colony mats that we get. But the problem is, remember, go back to why did we put these all under the category of can be? We put these all under the category of can be because they all looked the same. And there wasn't a way to morph them, based upon colony or microscopic appearance as being, different genera or different species. And so you can do downstream diagnostics from this product, by taking the colony and doing speciation through molecular techniques. But just having this grow up cannot tell you that this is Nenisiopsis or Paranenisiopsis or a species after that. Histopath has been, indicated that this is the gold standard. And I agree, you want to be able to say, there is fungus and it is in the lesion. However, it does require biopsy and anesthesia. You can use special stains to confirm that fungus is in the tissue. Our pathology partners love showing us these beautiful pictures. So here is an image from the literature. The big purple and all of the little purple spots in here are special stains to help identify fungal agents. But again, this cannot tell you this is N. goroi versus N. arthrosporoides. This can just tell you, hey, there's fungus and it's causing this lesion. Wood's lamp is something that's commonly used. We know that the white nose syndrome fungal pathogen of bats shines well with Wood's lamp. We know that some Microsporum dermatophyte species shine with Wood's lamp. I will show you in our data set. Nenisiopsis goroi, at least the isolate that we use, does not shine with Wood's lamp, but it is something that we have used and it's kind of ingrained as like an easy peasy, try it out in practice sort of thing. So I put it up here. But it doesn't work in my sample set. Panfungal PCR. So here we are getting into some molecular techniques. We can take a lesion sample, we can take a biopsy sample, and not all PCRs are built the same. Different PCRs are going to have different target. A panfungal PCR is looking at highly conserved portions of the fungal genome. Typically, this is ITS1, ITS2. This allows you to say, hey, there's a product, there's a DNA product that was able to be amplified in the sample that you sent. It will not tell you what that product is unless you do downstream testing, which is typically sequencing in comparison to libraries of known sequences. So if we kind of look at pros and cons of each of those detection diagnostics, I kind of break these into, can you do them in-house or do you have to send them out? Are the results immediate or do you need to wait a little bit? Is anesthesia required? What's the approximate cost? And is there any other rule in, rule out capabilities, right? And so cytology becomes really attractive because it's in-house, it's potentially in-house. Hopefully I've inspired you to do it in-house. Pretty immediate results, typically no anesthesia required. Low cost to you, low cost to client. And because it can rule out other things like a bacterial secondary infection, around cell neoplasia, as I showed you an image for, it really allows you to have a more broad strokes at your, figuring out a diagnosis for your patient. Things like histopath are not going to be in-house or immediate, but and do require anesthesia and are going to cost more, but also allow you to rule in and rule out. That's going to be the gold standard for ruling in or ruling out other differentials. So hopefully this can help you kind of have some of those conversations with your client about, what's the biggest bang for your buck? Is this fungus and, kind of, or is this something else, a neoplastic condition that's there? From there, if we get that, yeah, I'm not just my spidey senses are tickling, not just that this fits a clinical presentation of a mycosis, but also that I've detected fungus by one or several of the following, of the prior diagnostics, then we have the opportunity, and I would push you to try to have our clients identify what is the fungus that's there. And we have a couple of opportunities here. The first one is a quantitative PCR. The next one would be MALDI-TOF. MALDI-TOF is kind of challenging to explain. If PCR is looking at DNA. MALDI-TOF is looking at proteins, so proteinomics. And so this is done at a lot of our specialty laboratories now, where they can take, that growth on an agar plate, whether that's bacterial growth and sometimes fungal growth. They can take a little bit of the colony, put it in this fancy machine, and it's basically doing mass spec on the on the proteins that are there. And they compare the proteins that are there and in which quantities to known libraries, so proteinomic libraries, to try to get a diagnosis. And this has been shown to be able to work for N. guaroi, but we really have pretty limited fungal proteinomic databases at this time. So there's always kind of like a little asterisk next to diagnoses via MALDI-TOF with fungus right now. And then that moves us to NGS, or next generation sequencing. So this is the MyDog technology, and this is why we're here today. Thank you, MyDog, for hosting us. This is kind of a screenshot of some of the results that you get from MyDog, where what the MyDog technology is doing is truly doing these big consensus PCRs for both bacteria and fungus. So trying to amplify all of the DNA for bacteria and fungus in the sample that is sent, but then it does an immediate downstream sequencing of all of those amplicons or amplified pieces of the DNA. And after sequencing, comparison of those sequences to known databases, where we know that N. guaroi has this sequence in this area of the genome. So these are truly our identification tests that we have available. A lot of people ask me, why should I care about fungal identity? They say, there's a bearded dragon, identified, that it has fungus on it. Who cares if it's N. guaroi versus N. arthrosporoides? And so I present this to everyone. So here's case number one. You use all of the diagnostics available to you, and you're able to get to a diagnosis of genus and species, where you have a confirmation that you have N. guaroi. Well, if you have that, there's a whole lot in the literature that you can use to drive therapy, and a growing amount in the literature to drive therapy. And so that can be of huge benefit to you creating these therapy protocols, to your client to understand, okay, there is this information out there, and to your patient, to really modify, what we have available for them. Case number two, we used all the same diagnostics, and we know that it's something besides N. guaroi. So maybe we get to N. arthrosporoides. Well, there's a lot less literature on N. arthrosporoides in terms of MIC data, treatment data. And so we can't use all of the literature. And that would push me to counsel a client to potentially order MICs. Right? So I would say, okay, we have this culture, we've used this culture and done this and done that. Why don't we order MICs for terbinafine to see if we can modify a treatment plan based upon those MICs? Because we really don't have that in the literature. And then case number three is probably everyone's bane. You know via cytology or your spidey senses, this is fungus, and it's probably Nenisiopsis or Paranenisiopsis, but they do not approve anything outside of that diagnosis. And they're asking you to set up a perfect treatment protocol that will cure their, chameleon stat. And so that's when, it's like, well, if our clients want us to be magical, we need to be able to consult all of the information that guides our magical capabilities. And so, kind of really setting up client expectations that we are, in fact, not magical. And, we do need some more information to help guide them. And then the last thing is that, we do know a lot more about, cutaneous mycoses in reptiles than we did 10 years ago and 20 years ago. Imagine what we can know in 10 or 20 more years if we continue to move the industry forward. If we can continue to say, this is how we treat N. arthrosporoides, or these are certain treatment considerations for N. arthrosporoides versus N. guaroi. And that requires on the ground moving it forward. And some of that is on our clients to help us move that forward. Us researchers are doing everything that we can as well behind the scenes. All right, so this is the question of all questions. Which diagnostic is the best diagnostic? And there is no right answer. I think we need to pair our diagnostics with the needs in front of us, right? If we are like, hey, is this cancer or fungus? We need to reach for a super basic diagnostic that's going to help us rule in or rule out other differentials first. If we have a super motivated client and they're like, I really don't care what we have to spend, but we need to get the best treatment needed for my beloved, whatever species is in front of you, then maybe we need to do a lot more diagnostics in order to utilize the literature more effectively. So, there's some wiggle room there, but I did ask this question of what is the best diagnostic? And this is a recent study that I did. This was my master's student, Dr. Amanda Wong. She actually just deposited her thesis yesterday at University of Illinois, so super proud of her. But our study aim was to look at the clinical sensitivity and specificity of diagnostic tests. Really, which of these diagnostics could help detect fungus throughout the course of Angora disease manifestation. And we hypothesized that those molecular diagnostics, both the qPCR and NGS, would have the higher sensitivity, clinical sensitivity, than some of the other diagnostics that we pursued. So there were a couple of adult bearded dragons maintained as a research colony at University of Illinois, where I used to work. They were systemically healthy. And they were maintained as normal bearded dragons should be maintained. So no abnormal care was applied to them. And they've been a research colony for 4.5 years on that site. So I told you there's a Cox postulates paper for Angora causing this yellow fungus disease in bearded dragons. So we already had this great mechanism to infect the bearded dragons. And based upon those bearded dragons, we should have infected the bearded dragons kind of on day 1, and then developed lesions kind of week 2 through week 4. And then we were going to carry them out a couple more weeks to see lesion development and allow us several weeks of weekly diagnostic collection. And then they were planned to be euthanized at the end of this study. So we were like, great, 6-7 week study, we got this. The plan was a cutaneous exposure to Nenisiopsis goroi. That was literally, there was no escarification, there was no injection. It was using a pipette and putting a couple droplets of basically Nenisiopsis soup. So fungal canidia, really just sitting in a water bath. It was allowed to dry over their backs. And this is pretty much what that past paper did as well, is just exposed the bearded dragons to canidia on their intact skin. The plan for weekly collection was to do cutaneous body swabs. So we were using these swabs and kind of rubbing all over the dorsum of the animal, all over the ventrum of the animal, to really just do kind of a global assessment of, what is there? Can we pick up this fungus? When they developed lesions, we swabbed the lesions directly, as well as did this whole body swab. And those body swabs were submitted for NGS through my dog, for fungal culture, and for qPCR for detection of Angora. We did pursue cytology, but we only looked at cytology once they developed lesions. So once there was a little scab, we would then, collect cytology to confirm, like, hey, this lesion is truly from fungus. So it was more of a confirmatory test and less of a diagnostic that we were assessing within this model. And we also took woods lamp pictures of them. I already gave you the preview that it doesn't work, but we have a whole bunch of beautiful, glorious photos of woods lamp illuminated photos of bearded dragons. And then we did some body mapping, and at the end of the study, they were euthanized, and we collected a couple of different cutaneous lesions from them with a complete necropsy. So remember I said it was supposed to be a 6-week study. Well, all good science goes through growth spurts. We did infect the bearded dragons on day 0, and by 4 weeks, which is when we should have had all the bearded dragons develop clinical signs, we had 0 bearded dragons develop clinical signs. So at week 5, we were like, maybe we should expose them again. So we concentrated the exposure, made it a thicker soup, and gave them some more exposure. By week 9, so another month later, still no lesions. We're not panicking at all. We again repeated the exposure with that more concentrated kind of soup of Angora canidia. And then, finally, week 11, we have our first lesion that popped in a male. By week 13, just kind of 4 weeks after the last exposure, we had another animal pop with a lesion, and that was another male animal. And then week 14, we had 3 more animals that were positive, 2 males and a female. We repeated exposure for that one that did not have lesions. And then finally, at week 17, the very last holdout female was positive for her lesions. And then we brought them out to week 21, and at week 22, they were all euthanized. So we had originally planned that we would do weekly data collection where we would do culture and Woods lamp, NGS and qPCR. But when 6 weeks turns into 21 weeks, that's a whole lot more data collected. All of the data was collected, but in terms of financially prioritizing how to represent the longevity of the data, what we really had to do is prioritize putting in and assessing samples of monthly NGS, so my dog and qPCR analysis. And so this makes our results a little bit imbalanced because we do have weekly culture results, but we only have monthly NGS and qPCR results. And that's just because the length of this study became a little bit of a bear. Again, on postmortem, we did these body maps, just kind of identifying where each of the lesions were. And we kind of arbitrarily took 2 to 3 lesions per lizard. Each lesion was tested for qPCR, cytology, Woods lamp, histopathology in that postmortem area as well. We looked at the time to initial positive, and I'll show you kind of how we assess that number. And we just applied some basic non-parametric comparisons to those time to first positive. So I'd have to say, we had some unexpected findings with our results that I'll share with you. And then I wanted to talk to you a little bit about both the antemortem diagnostic performance as well as the postmortem findings that we had for these animals. So, I kind of gave you a preview in the intro, but the lesion development, we put the fungus on the back of the animals. And although we were kind of potentially rubbing that fungus everywhere over their body every week when we were doing our data collection with those body swabs, we did not expect that all of the animals would develop their first site and most incredible site of infection being in their femoral pores. And this presented initially with just kind of what looked like an impacted pore. So this was one of the males. You can see the pores are pretty large, larger than they would be normally, maybe a little bit of impaction here, but there would be like one pore that would just look a little red around it. And it wasn't, that first bearded dragon that popped with infection, I literally looked at him and I was like, oh, you have this impacted pore. I think you need this popped out because I'm a picker and this is a dermatologic lesion, but it really wasn't me considering, hey, I think this is going to be fungal induced. So we popped it out. We rolled out that kind of little waxy plug on a slide, and I was expecting them to come back cytologically as keratinaceous debris or maybe some bacterial infection. I was not ready for them to tell me that this was the very first instance that we had of, fungal infection. And this wasn't one bearded dragon. This was all 4 of our male bearded dragons. This is where their infections started. So just really interesting that it was on the ventral aspect of their body and all associated with these femoral pores. Both of the females did develop lesions. They developed them a lot later. Their lesions were a little bit more traditional, but they were mostly on the ventrum. So this is on postmortem of one of our females. You can see she has kind of a dark crusty lesion and a more traditional yellow lesion. Both of these lesions are proven to be from fungus. So just another kind of pusher that the lesions are not always yellow, and they're not always yellow on the same patient, you know? So, and truly none of the lesions developed at the exposure site, which is, right on dorsal midline for these animals. The other unexpected result was this real discrepancy between the female bearded dragons developing lesions and the male bearded dragons developing lesions. So just to kind of show you, we had small numbers here. We really only doing this with 6 bearded dragons. So we had 4 males and 2 females. But those 4 males, were 78 to 99 days was their time of first lesion after that first initial exposure to the fungus. And so that's a median date of 96 days. Our females were a lot later, up to 120 days later. None of the females had those lesions on the femoral pores. And it was just a very unexpected sort of finding that I think deserves more digging, like in the future, how do we figure out what is the difference here? Or was this just a fluke thing because we have small numbers in this study? When we talk about our antemortem diagnostics, none of the lesions fluoresced under Wood's lamp. So here is one of our female bearded dragons. She has a lesion where each of these arrows are. And you can see from the image at the very same time, she does not have significant fluorescence at any of those sites. That's not to say we didn't get a lot of beautiful pictures of fluorescing. I'm going to shed skin soon. That fluoresces real nicely. Here are the pores of a male. These are the normal pores of a male. Those fluoresce nicely as well. Urates, so if they have a little bit of urate contamination on the tail, those look nice. And then any calcium carbonate. So if they had a little bit of calcium around their lips, that would also fluoresce. So we had lots of pretty pictures, but none of them were helpful in detecting fungus. So if we look at each of our individual animals here, so we have kind of these brown lines, which represent the male animals, and the green timelines are representing our female animals. The days here are the days when there's an E, which is the repeated exposure that these animals needed to have. And then the boxes are when each of these animals had their first initial lesion. So in order to really assess our diagnostics in the face of this non-uniform time to initial lesion development, what we did was really made all of the animals' initial date of lesion development to be kind of this new time zero, okay? And so I'm just going to show you when the first detection of fungus happened with our 3 diagnostics kind of surrounding this initial lesion development. So there's that initial lesion development, kind of a new time zero for these animals. So our first positive culture preceded lesions in all 6 of the cases, okay? So you can see that is highlighted here in yellow for you with the days that was positive. The first positive NGS, my dog sample, proceeded in 5 of the 6 cases. And so here it is in blue, showing that all 5 of these animals were able to detect the fungus before the date of lesion development. And then qPCR was a little bit different. So I apologize, the color scheme changed on me when I changed this, but it's at 93 days here. So before on this one, it's at 93 days here at the same time as this one, 93 days here after this one, before this one, before this one, and before this one. So a lot of discrepancy between when these first initial positive diagnostics kind of came through. If we look at that diagnostic performance between them, we can see the number of positive tests that were positive for fungus, Nenisiopsis, prior to developing the cutaneous lesions, at 100% for culture, 83% for my dog, and about 2/3 for qPCR. But we're running with some very small numbers, right? We're only talking about 6 animals. The other thing that I want to point out is that NGS, or my dog, and qPCR didn't get their fair time in the sun because we only have monthly evaluations rather than weekly evaluations, which we have for culture. And so, there is a lot of question there surrounding, there is a skewing of these results to favor culture being positive earlier. And then the last kind of finding would be, something from our postmortem results. We had 6 bearded dragons that we experimentally infected, and all of them developed this multifocal dermatomycosis, some of them taking a lot longer than others. But I think more importantly for these animals, that 1 of 6 of them developed systemic mycosis. So one of the bearded dragons did have a pulmonary granuloma that was proven to be N-Groy on necropsy. This animal had no respiratory signs. He didn't seem sick in comparison to the others outside of having these lesions. Whoa, I got some balloons going for you guys, sorry. So it's I think we need to keep this in mind again as we think about, how these fungal infections can manifest in patients. So lesion development was pretty prolonged in our study compared to, that prior study, which is this Gentry and al. And what are the reasons for that? We can only postulate, but we did use adult immunocompetent bearded dragons. Maybe next time I would follow suit with the past study and use some younger bearded dragons that maybe, weren't quite so immunocompetent. We did not abrade the skin. The prior study also did not abrade the skin, but in some of the animals, they did abrade the skin. They just found that it really wasn't necessary for them to become infected. And then the dose given. So we did give much higher doses. Even our initial dose given was around 3.5 million canidia per lizard, whereas they gave 2 canidia per lizard. We did both use N-Groy, but we were also using very different isolates of N-Groy. And so, is there a difference due to the Nenisiopsis isolate there as well? And then initial lesion development was obviously very, 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 very different. Lesions that did develop were different. However, that Gentry paper did not report on any femoral pore development, and that could be a component of age as well. If our adult male bearded dragons have more, blood supply to their pheromone producing femoral glands, maybe that's a reason why it was a more active site for infection. And, to really dissect, why disease presentation could or would differ by sex, I think there's a lot of questions I have here. Obviously there's a question around, this host difference. And I think that that's something that we need to explore, and I am exploring in the future, because it's really going to pertain to, our patients' outcomes potentially. So. Because these animals did have identical environment. Between male and female, we had an identical microbe that was used. And so that really does mean that there needs to be a host difference that drove these differences. So in the end, we have these couple of black spots on this bearded dragon. This is from the infection trial. These are some of the very earliest pictures of what was abnormal in these bearded dragons. How do we know if these black spots are N-Groy? So do not use wood lamp, or if you do use wood lamp and get some fluorescence, we only tested 1 isolate. So even with dermatophytes, it's only like 30 or 50% of microsporum isolates are going to fluoresce. So please let me know if anyone is seeing some fluorescence, and we'll dive deeper, but I wouldn't use that as a huge criteria. Fungal culture, not super expensive, but it's going to take a while. it is a detection test if all you do is fungal culture. And if you want to know Nindisiopsis or Groi, you're going to have to do use that product, which is the colony for follow-up speciation, which is going to be additional money, additional time before you get the diagnosis that you want. It is able to detect other fungal diseases, but it's definitely not able to rule out that this is cancer, right? So just kind of a point there. Quantitative PCR, I was part of the team who developed this PCR. It goes through the wildlife epidemiology at U of I. It takes about a week, a little bit more expensive than fungal culture, usually around $100 plus shipping. It is Nindisiopsis consensus, so it can pick up Nindisiopsis DNA. It can't tell you if it's N. Groi at this time. And so if you wanted to know if it was N. Groi, it would really have to also do some downstream speciation with sequencing. And it's really not able to detect any other bacterial or fungal causes. So before you are hitting up a fungal culture, before you are hitting up a qPCR, you should do something else in office or out of office, either a biopsy or a cytology to say like, hey, this is fungus, and I'm going to rule in or rule out which fungus this is. And then we have the MyDog next generation sequencing. I put around a week, but it's not. Thank you, Janina, for saying that at the beginning. It is a rapid turnaround time. And to hear that you guys are going to move to 24 hours is bananas and incredible. Not bananas in a bad way, bananas in a very, very good way. Going to be potentially the most expensive with shipping included. And I don't think that I have the correct price there. I apologize. But because it is able to pick up both bacterial and fungal DNA and detect that DNA, it is going to be able to rule out other infectious causes of this presentation. So it gives you a little bit more flexibility and definitely a much faster turnaround time. I put sterile sample collection. You want to do clean sample collection. So because we don't want your DNA in there, sort of thing. And yeah, yes, able to detect other bacterial or fungal causes that can cause this. And, we've been harping on Paranindisiopsis and Nindisiopsis, but this could be Fusarium. It really could be. And so there are other microbial presentations that maybe aren't as classic that could definitely cause what we see to be the kind of this yellow fungus disease. So just a couple of little things, there's a long way we still need to go with Nindisiopsis. We need a case definition. We need to look at diagnostic performance in natural infections. That's something that we need to look at. We need to look at that influence. What is the difference between male and female, and why did we see that? And we definitely have study limitations. It was 6 animals. We have an imbalanced study design with not all diagnostics pursued at every time point. And we have a pretty limited statistical analysis of both the antemortem and postmortem diagnostics. Just wanted to throw this out there. I do have a ton of references. That's on a Google Drive if anyone is interested in stealing those references. Super easy peasy. This was not a project that I did. This is a project that I got to work on. Awesome team. We had a whole dragon team that took care of the animals at U of I, a bunch of students. I got to hang out with clinical pathologists and anatomic pathologists. Laura Adamovich is at the Wildlife Epi Lab and does a lot of work there. And of course, I got to work with MyDog as one of the first times that I was working with MyDog is in conjunction with this project. So that's what I have for everyone today, and happy to answer any questions about anything we talked about.
Thank you so much, Krista. That was a wonderful presentation. We do have a few questions in the chat. So one attendee is asking, what sample type is best for these cases? Just a swab or a scab, debris, tissue, biopsy? What would you prefer?
Yeah, I think it's according to what you're going for in terms of sending for qPCR, MyDog, or fungal culture. The more DNA you can collect, the more likely the molecular diagnostics are to pick up these pathogens. we want to try to decrease the amount of feces in the sample, which some of your patients and some of the lesions can be around the areas that have a lot of feces, because feces can have some prominent inhibitors. But in general, if there's some chunky, like skin coming off with it, that can be really helpful. All the animals for this experimental data set, they had body swabs. So they did not have chunky monkeys that were sent or on. It was just what skin flakes were coming up with a moderate pressure applied to the swab over the skin. So we can get a diagnosis without doing a biopsy. We can get a diagnosis without sending in crusts, etc.
Okay, then we have another question. I think it was asked while you were presenting, so maybe you answered part of this already. The question was, were these lizards roughly age matched? If so, what age? Do you often see this presented in young bearded dragons, or is there a similar presentation or more rapid progression based on the age?
Yeah, all of the bearded dragons that we infected were the same age. They were obtained at the same time from the same breeder, but they were not from the same clutches. So they may have been different in age by maybe a couple of weeks at most. I see Menisiopsis infections in young and old and everything in between. I'd say that the hardest cases are probably the older animals, but it's usually because they have other comorbidities. And so I'm not sure that it has to do directly with the microbe, but maybe that they have chronic renal failure on top of it, and how we have to treat them is not helpful in that chronic renal failure. Or they have hepatic lipidosis, really, really common in older bearded dragons, and they're not really tolerating, an azole therapy. So, I don't know if that answered the question, but I don't see a huge difference in clinically in the cases that I'm seeing due to age.
Okay. Yeah, we do also have a couple of compliments for your talk. Thanks you so much for the informative lecture. People are glad to see you back at UC Davis. The next one, what is the actual cost of MyDoc NGS testing? And we are at $155 per sample, which does include shipping, FedEx overnight to our lab, and free veterinary consultation. Will this presentation be made available? Yes, it will. We will send out a recording afterwards. Dr. Aguilar also says, thank you for the amazing presentation, Dr. Keller. And I believe that is all the questions that we had so far.